Pepsin is a proteolytic enzyme that begins the process of protein degradation in the stomach. Pepsin action yields protein fragments that will be further degraded by the proteases of the intestine. Due to the environment of the acidic stomach, pepsin displays optimal activity in the pH range of 1 to 2. Pepsin functions best at these pHs because the carboxylic acid group on the amino acid in the enzyme state must be protonated. Pepsin is one subunit that consists of 326 amino acids. Highlighted in yellow are two important amino acids. The interaction between aspartate 32 and aspartate 215 leads to the formation of the active site. The secondary structure shows 10 alpha helices encompassing 46 residues that are shown in pink and 32 beta strands encompassing 142 residues that are shown in yellow. The turns are indicated as well in white. Secondary structure is crucial in achieving enzymatic function. Two antiparallel beta strands on the surface of the protein around the residues threonine 77 and glycine 76 form a loose flexible flap. The flap is located near the cleft of the active site, and its high degree of mobility is hypothesized to allow substrates into and out of the active site, which can shield the active site from the environment while the reaction proceeds. The flexibility and stability shown in the secondary structure lead to pepsin's form and function. Here the flap is shown in this green color with these two antiparallel beta strands covering the active site. Pepsin is classified as bilobal, or having two lobes or domains, which become close in proximity upon proper protein folding and is critical for the formation of the active site and its biological function. The active site of the acid proteases rely on two acidic aspartate amino acids, which activate a water molecule and use it to cleave protein chains. One of them is shown here in the active site, and the other one is shown here. Also shown here in the tertiary structure are these green highlighted areas, which are disulfide bonds that are formed between the sulfur atoms and cysteine residues, and their function is to strengthen the folded protein. The specific reaction catalyzed by pepsin is the acid hydrolysis of the peptide bond. This reaction will break down proteins into smaller units to enable the digestive process. Pepsin catalyzes the hydrolysis of peptide bonds between hydrophobic or aromatic residues of a protein substrate. Bonds involving phenylalanine and leucine are commonly cleaved. This slide shows the arrow pushing mechanism. The acidity of aspartate residues in the active/catalytic site functions to cleave the targeted peptide bond of the substrate through a mechanism of general acid-base catalysis. Water plays a crucial role in this mechanism. Glycine 76 and threonine 77, shown in this ball and stick model, are part of the flap that anchors the substrate near the active site through hydrogen bond formation. The polar nature of threonine, due to the free hydroxyl group, contributes to these hydrogen bonds. The area that is circled in white is the active site, as well as the catalytic site. In this specific enzyme, they are the same. This structure also includes a non-cleavable inhibitor in the active site, which is shown in orange. This could be a representation of pepstatin, which is a specific inhibitor of acid proteases that binds tightly to pepsin. It contains a hydroxyl group, shown here in purple, that mimics the place that water attaches during the cleavage reaction. Water would form a bond with the aspartate residue that is deprotonated, and therefore pepsin does not actually form chemical bonds with the substrate. This was shown earlier in the arrow pushing mechanism. More importantly, in the stomach, protein chains bind in the active site of pepsin to be cleaved. Just to reiterate what I said before, the catalytic site of pepsin-like enzymes is formed at the junction of the domains and contains two aspartic acid residues, aspartic acid 32 and aspartic acid 215, one in each domain. Pepsin is an aspartic protease using a catalytic aspartate in its active site. In conclusion, pepsin is an important enzyme for the digestion of proteins, where two residues, aspartic acid 32 and aspartic acid 215, are mostly responsible for cleaving the peptide bonds. The flap created by antiparallel beta strands near the active site is responsible for keeping the substrate from the environment and aid in the acid hydrolysis reaction. If we did not have pepsin, our bodies would be unable to digest proteins. One small scientific note is that the small intestine releases bicarbonate to buffer the acidic pH of pepsin. The pH is about 8, which is way too high for pepsin, and thus pepsin is denatured in the small intestine.